All right, today we're diving into what is basically a real-life detective story, but it's happening at the molecular level. We're looking at this really baffling case in colorectal cancer where, get this, the cure can actually backfire. And the lead detective on the case? It's a high-tech AI that's helping scientists finally crack it. So let me ask you something that sounds like it's straight out of a thriller. What if the very drug that's designed to stop cancer actually ends up making it stronger? I know, it sounds crazy, right? But this isn't some hypothetical scenario. It's a real, genuine mystery that has had researchers scratching their heads for years. And that's exactly the case we're cracking open today. Okay, so to really get our heads around this mystery, we first have to meet our primary culprits. Now, these aren't people, of course. We're talking about two very specific gene mutations. You could almost call them the masterminds behind this really tough form of cancer. And here's the thing. Our two main suspects are incredibly common. I mean, look at this. The KRAS mutation, that shows up in about 40% of colorectal cancer cases. And BRAF, that's in another 10%. So you do the math. Together, these two are involved in roughly half of all cases. That makes them absolutely critical targets in this whole investigation. So what do these genes actually do when they're behaving themselves? Well, you can think of them as key players in a cell's internal communication network. KRAS, that's the regulator. It's basically a little on-off switch for cell growth. Then you have BRAF, the messenger. It takes the growth signal from KRAS and passes it down the line. In a perfectly healthy cell, they're a perfect team. But, of course, in cancer, things go wrong. These genetic masterminds, they don't just break the rules. Oh no, they completely rewrite the playbook. They hijack one of the most critical pathways in the whole cell and turn what should be a regulated process into an absolute runaway train. And right here, this is our crime scene. It's called the MAPK signaling pathway. The easiest way to think about it is as the cell's main command chain for all things growth. It's the series of signals that literally tells a cell, okay, time to divide, or nope, time to stop. And that process, as you can imagine, is absolutely essential for everything to work normally. So let's walk through how this is supposed to work in a perfect world. First, a signal comes in from outside the cell, a little message that says, hey, it's time to grow. Then KRAS, a regulator, flips to the on position. That in turn activates BRAF, our messenger, which carries that signal onward. The end result? Normal, controlled cell division. It's a nice, orderly, super-regulated chain of command. But here's the flip side. Here's what happens when things go haywire. When KRAS or BRAF get mutated, they get stuck. The switch is jammed in the on position. The technical term for this is constitutive activation, which is just a fancy way of saying it's always on. This means they are just constantly screaming, grow, 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 down that pathway, even when there's no signal telling them to. And the result, as you can guess, is that uncontrolled cancerous growth. Now, hearing all that, you'd think the solution is pretty simple, right? Just create a drug that blocks that faulty BRF protein and shuts the whole thing down. And, well, scientists did exactly that. But this is where our whole investigation takes a massive plot twist. Because those first attempts to stop the bad guys, they had this really bizarre and frankly dangerous side effect. Yeah, this quote right here from the research just sums up the whole problem perfectly. The drugs that were specifically designed to inhibit BRAF could sometimes do the complete opposite. It's a phenomenon that scientists call paradoxical activation. The name says it all, really. So what does that actually mean? It means that under certain conditions, especially if that other gene, KRAS, is also mutated, giving a patient a BRAF inhibitor drug could accidentally kick the entire growth pathway into overdrive. The very treatment designed to stop the cancer was, in a twisted way, helping it. I mean, you can imagine, this was a massive, massive setback for scientists and a huge problem for patients. Okay, so the case is getting more and more complicated, right? So the cancer detectives, they started to think, maybe we're looking in the wrong place. They began to wonder if the problem wasn't just what was happening inside the cancer cell, but what was going on in its surrounding neighborhood. And that line of thinking led them to something called the tumor microenvironment, or TME for short. This is basically the whole ecosystem that a tumor lives in, the blood vessels, the immune cells, all the stuff surrounding it. And a new theory started to form. Maybe, just maybe, another cell in this neighborhood was secretly acting as an accomplice. And that, my friends, brings us to a brand new suspect, the neutrophil. Now, neutrophils are supposed to be the good guys.
They're a really common type of immune cell. But the hypothesis was this. What if the neutrophils hanging out in the tumor's neighborhood are actually releasing substances that modify that mutated BRAF protein? And what if that modification is exactly what's helping the cancer cells resist treatment? It's like a trusted cop secretly helping the bad guys.
Okay, let's just recap for a second. We've got mastermind genes, a hijacked pathway, a cure that can backfire, and now a potential surprise accomplice. This case is getting incredibly complex, right? So to make sense of all these clues, the investigators decided it was time to bring in a powerful new partner. Enter the Gaia Lab AI platform. I want you to think of this thing as a super detective. It's an AI that can sift through absolute mountains of data. We're talking genetic data, research papers, clinical trials, and spot these tiny patterns and hidden connections that a human researcher might just miss. So its job was to analyze all the evidence and see what it could find. And the AI delivered a major lead. Fast. It flagged an existing, already FDA-approved drug called Sorafenib, giving it a 76% match score for treating this specific cancer. Now, the reason why is so key. It doesn't just go after BRAF, it targets multiple culprits at the same time. And that strategy could be the key to sidestepping that whole paradoxical activation problem. You know, the implications of this are just huge. Repurposing a drug that's already out there is way faster and way, way cheaper than developing a brand new one from scratch. The AI's analysis estimates that this kind of approach could save up to, get this, $1.9 billion in development costs. But what about our surprise accomplice, the neutrophil? Well, the AI weighed in on that too. It analyzed all the evidence for that hypothesis and came back with a 55% consensus signal. Now, what that tells us is that the neutrophil link is a really promising lead for sure, but it's still considered exploratory. There just isn't enough hard evidence yet to close that part of the case. And this is the real power of the AI. It didn't just give us one lead. No, it generated a whole lineup of suspects. Check this out. These are the top drug repurposing candidates it found. Every single one is already FDA approved, they all target the key mutations we've been talking about, and they're all potential new weapons in this fight. So you've got these hot new leads from your AI detective. What's the next step? Well, the investigation now moves from the digital world back into the real world, back to the lab. Scientists now have to run the experiments to see if these digital clues hold up and hopefully, finally, close this case for good.
And here's exactly how they plan to do it. It's called a perturbation assay, but you can just think of it as a sting operation. Step 1, you take cancer cells that have that BRAF mutation. Step 2, you introduce the main suspect, the neutrophils. Then you just watch and measure what happens to the cell's activity. If the neutrophils really are the accomplices, scientists should see a clear shift, a move towards that drug-resistant state, and that would be the proof they need.
And it's so important to remember why all of this detective work actually matters. This isn't just some academic exercise to get published in a journal. Every single clue they uncover, from figuring out that paradoxical activation to nailing down the neutrophil link, every piece of the puzzle is a step toward developing smarter, more effective, and truly personalized therapies for real people. Which really just leaves us with one final tantalizing question, doesn't it? Could it be that by combining the power of an old repurposed drug with the incredible intelligence of a new AI, we're on the verge of finally solving one of the toughest cases in colorectal cancer? All the evidence seems to be building, and it feels like the detectives are finally closing in.